Welcome back to the deep dive. Today we're doing something a little different, a highly targeted mission.
Yeah, we're going deep on just one single paper.
Exactly. We've taken one of those, really dense but important papers from the New England Journal of Medicine, and our mission today is to unlock what it really means for a cutting-edge cancer treatment.
And it's the kind of evidence that doesn't just, tweak the standard of care. It can really change the entire playbook for patients at a very critical point.
That's the key. So we're talking about a drug called Sacituzumab Tirumotecan, or SAC-TMT for short. And to really get why this matters, you have to understand the patients we're talking about.
That context is everything. We're focused on non-small cell lung cancer, NSCLC, which is already a huge challenge, but this is a very specific subgroup.
Right.
We're looking at patients with an EGFR mutation. Now, that mutation means they initially respond well to targeted therapies, but we're looking at the point where those drugs have stopped working.
So their first line of defense, that EGFR tyrosine kinase inhibitor, or TKI, has failed. The cancer has outsmarted it.
Precisely. And these patients have often already moved on to the next step, which is usually platinum-based chemotherapy. So they're in a tough spot. Their options are running out.
The toolbox is starting to look pretty empty.
It is, and that's where this study comes in.
So this is where SAC-TMT steps onto the stage. Let's unpack the drug itself. That name, Sacituzumab Tirumotecan, is a bit of a mouthful. What exactly is it?
Well, what's so fascinating is that it's part of this whole new class of drugs. It's an antibody-drug conjugate, or ADC.
Okay, the guided missile analogy, right?
That's a great way to think about it, yeah. Let's break down the name. The sacituzumab part, that's the antibody. Think of it like a GPS system.
So it's a targeting component, and what's it targeting here?
It's designed to find and lock onto a protein called trophoblast cell surface antigen 2, or just TROP2.
TROP2, got it.
And this TROP2 protein is found in really high amounts on these specific lung cancer cells, so the antibody just homes in on it.
So that's the delivery vehicle. Then what about the Tirumotecan part? That sounds like the warhead.
That's exactly what it is. It's the payload. Once the antibody attaches to TROP2 on the cancer cell, the cell actually pulls that whole complex inside itself.
Oh, interesting.
And that's when the tirumotecan, which is a really potent chemotherapy agent, is released directly inside the cancer cell.
So you're not flooding the whole body with chemo, you're delivering the poison right to the target's doorstep, or I guess right into their living room.
Exactly. You spare so much healthy tissue that way. It's precision toxicity.
And the paper mentions that this drug had already shown promise in smaller studies, which is why they launched this big phase 3 trial.
Right. They needed to prove, on a large scale, that this clever mechanism actually translated into a real, undeniable benefit for patients compared to the current standard of care.
Okay, so that sets the stage perfectly. Let's pivot to the trial itself. How did they set this up to prove it was actually better? The methodology is everything if we're going to trust the results.
For sure. Well, they kept the patient group very specific. Just like we said, EGFR-mutated NSCLC that had already progressed after TKI therapy.
And they weren't comparing it to a sugar pill, right? This was against the active established standard.
No placebo here.
Yeah.
This was a head-to-head fight. They took 376 patients and randomized them 1-to-1.
So half and half, 188 patients in each group.
Yep. 188 got the new drug, SAC-TMT, by itself.
And the other 188.
They got the standard of care for this situation, Pemetrexed plus a platinum-based chemotherapy. This is the direct comparison that makes the results so powerful.
Okay, so a fair fight. Now, what were they measuring? What were the endpoints?
So the primary endpoint, the main goal, was progression-free survival, PFS.
And that's exactly.
It's the length of time that a patient lives without their cancer getting any worse. And really importantly, it was assessed by a blinded review. The doctors reading the scans had no idea which drug the patient was on.
Removes any bias. Smart. And what about the big one? Overall survival.
That was the key secondary endpoint. And the methodology here is, it's really important. It was hierarchically tested.
Hierarchically tested. What does that mean in plain English?
It's a way to ensure scientific honesty. It means you can't even formally test for overall survival unless you've already proven a significant benefit in your primary endpoint, in PFS.
So you have to clear the first hurdle before you can even attempt the second one.
Exactly. It stops researchers from, just fishing for a positive result somewhere in the data. It adds a ton of credibility.
That makes a lot of sense. And they followed these patients for a good while, right?
They did. The median follow-up was almost 19 months, so this isn't a short-term snapshot. This is durable data.
All right, the moment of truth then. the results. Did the guided missile actually beat the old school chemotherapy?
It didn't just win.
Yeah.
The difference in progression-free survival was, well, it was massive.
Okay, let's start with the standard chemo group. What was their median PFS?
For the standard chemo group, the median time before the cancer progressed was 4.3 months.
4.3 months, okay. And for the SAC-TMT group?
The SAC-TMT group had a median PFS of 8.3 months.
Wait, say that again? From 4.3 to 8.3. That's basically double.
Essentially double the time without the disease advancing.
In this patient group, where options are so limited, gaining an extra 4 months of stability, that's not just a statistic. That's family events. That's quality of life. That's huge.
It's a monumental shift, and the statistics back it up completely. The hazard ratio was 0.49.
Which means?
That means about a 51% reduction in the risk of the disease progressing or death for patients on SAC-TMT. The p-value was less than 0.00001. It doesn't get more definitive than that.
So they cleared that first hurdle with flying colors, which means the door was open to look at overall survival.
Correct. And the analysis there showed that SAC-TMT patients also lived significantly longer overall.
What were the numbers on that?
The hazard ratio for death was 0.60. So 40% reduction in the risk of death compared to standard chemo. Again, highly statistically significant.
Let's make that tangible. Is there a landmark like a 1-year or 18-month survival rate we can look at?
Yes. The paper highlights the 18-month overall survival rates. For the standard chemotherapy group, it was 48%.
So just under half the patients were alive at a 1.5 years. And for the new drug?
For the SAC-TMT group, it was 65.8%.
Wow. So an almost 18 percentage point difference. That is a dramatic intervention.
It absolutely is. It translates to a large number of people alive at that mark who wouldn't have been otherwise.
That's incredible efficacy. But there's always a but. We have to talk about safety. A more powerful drug often means more powerful side effects. Was there a big trade-off?
This is maybe one of the most interesting parts of the study because ADCs are this hybrid of targeted therapy and chemotherapy.
Right.
So if you look at the overall rate of serious side effects, what we call grade 3 or higher, it was actually pretty similar between the two groups.
Yeah.
58% for SAC-TMT.
Okay, that's high.
It is. But the standard chemo group was 53.8%. So they're in the same ballpark.
And what was the main driver of those side effects?
It was mostly what we'd expect. A drop in white blood cells, specifically neutrophils, happened in about 40% of the SAC-TMT group versus 33% in the chemo group. Manageable and expected.
Okay, so the overall rate of severe but manageable side effects was similar. But I saw something in the paper that really jumped out. What about the really serious adverse events? The life-threatening stuff that lands you in the hospital?
This is the critical point. This is where that targeted mechanism really seems to pay off. When you look specifically at treatment-related serious adverse events, they were significantly lower in the SAC-TMT group.
How much lower?
It's a striking difference. In the SAC-TMT group, only 9% of patients had a serious adverse event.
9%. Okay,
Yeah.
and the standard chemo?
17.6%.
So nearly double.
Almost double the rate of these major, potentially life-threatening complications with the traditional chemotherapy.
That's a huge clinical win. You're getting a drug that is way more effective at controlling the cancer, helps people live longer, and it's less likely to cause a catastrophic complication.
That's the takeaway. It really validates the whole ADC concept. You might get some of the common chemo side effects, but you seem to avoid some of the worst systemic damage.
So let's synthesize this for everyone listening. What's the final verdict here?
The conclusion is crystal clear. For patients with this resistant EGFR-mutated lung cancer, Sacituzumab Tirumotecan provides significantly better progression-free survival and overall survival than standard chemotherapy.
It's not just a new option. It sounds like it's the new standard for this specific situation.
It should be. This is a landmark study. It gives clinicians a powerful science-backed tool to use when targeted therapies fail, and it's a tool that gives patients more time and potentially safer time.
And that leads us to our final thought for you to chew on. This study didn't just show higher efficacy. It showed higher efficacy with a dramatic reduction in the most serious, life-threatening side effects.
A critical combination.
So the question is this: how will this dual success force us to redesign the entire playbook? Is it even acceptable anymore to just default to traditional systemic chemo after a targeted therapy fails?
I think that's exactly the right question. This study sets a new bar. It suggests that this kind of targeted toxicity, like a TROP2 ADC, should probably be the next step before we even consider falling back on older, more broadly toxic regimens. That's the new standard this paper really sets for the future.
A huge step forward for precision medicine. Thank you for joining us for this deep dive. We'll see you next time.